In this video, I'll give an overview of how replication is initiated in eukaryotes and mention some essential mechanisms that regulate this process. To start, I want to briefly review the eukaryotic cell cycle, which is characterized by four main stages. In the G1 phase, the cell is growing and carrying out normal metabolic activity. In the S phase, DNA replication occurs. This is followed by G2 phase, in which the cell continues to grow and prepares for cell division. And then, of course, M phase or mitosis is when the cell divides into two daughter cells. In general, the progression through the different stages of the cell cycle is controlled by cyclin-dependent kinases. Specific cyclin proteins are expressed at different phases of the cell cycle, and the cyclins control which kinases are active in each phase. Replication is confined to the S phase of the cycle, and it's important that replication is completed before the cycle continues. Stalled replication forks prevent progression of the cycle. It's also important that replication is not initiated more than one time at any replication origin in a given cell cycle. Generally speaking, eukaryotes have much larger genomes than bacteria, spread over multiple chromosomes. So to complete the task of DNA replication in a reasonable length of time, we need multiple replication origins. The bright areas in this figure represent newly synthesized DNA. From 30,000 to 50,000 origins fire in a typical cell cycle in humans. It's hard to find eukaryotic replication origins just through sequence analysis, though there seems to be a preference for regions with low nucleosome concentration. To prevent an origin from firing more than once in a cell cycle, initiation is divided into two stages. Licensing, during which two helicase complexes are localized to the origin, and activation. The process of licensing is summarized here. An important group of proteins is the origin recognition complex, or ORC. This is a heterohexamer. MCM is another heterohexamer composed of subunits labeled 2 through 7. Two helper proteins called CDC6 and CDT1 are also involved. So the basic steps are listed on the left. First, the ORC complex binds to the origin. Then CDC6 binds. And then a complex of CDT1 and MCM binds, and the helicase is loaded onto the double-stranded DNA. The protein hexamer actually surrounds the double helix. Then an ATP hydrolysis event causes CDC6 and CDT1 to dissociate. A second CDC6 binds either at the ORC-MCM complex or at a second ORC that is bound nearby. CDT1 brings a second MCM complex, which is also loaded onto the DNA. The MCM complexes are touching each other at the origin. After the two MCM complexes have been loaded onto the DNA, the other proteins are not needed anymore, and so they can dissociate, and the origin is now said to be licensed. After being loaded, the MCM helicase just sit there at the origin throughout G1 phase, waiting for S phase to arrive. At the start of S phase, the kinases CDK2 and DDK become active and control the activation of the replication origin. The picture at the right shows an active eukaryotic replisome. If you look closely, you can see the MCM 2 to 7 hexamer here associated with CDC45 and something called the GINS complex. These other proteins are required for the helicase to be active. In S phase, the MCM complex is phosphorylated, which results in recruitment of these other proteins to the helicase, activating it. The other proteins required for replication, like the primase, the clamp loader, and the clamp, single-stranded binding protein, and DNA polymerase, are all recruited to the active helicase, and replication can begin. It's interesting that only about 20% of licensed replication origins are activated. One reason you might want other licensed origins in reserve, waiting to go, is if replication is stalled because of DNA damage. If one replication fork gets stopped, the cell could activate a nearby replication origin to allow replication of the DNA from the other direction. It is thought that as replication passes a licensed but not activated replication origin, the replisome simply pushes the MCM complexes off of the DNA. What the cell wants to avoid is licensing of replication origins during S phase. If this happens, then an origin could be activated twice during the same cell cycle, which would result in excess DNA. Several mechanisms contribute to preventing licensing during S phase. CDC6 is phosphorylated and is exported from the nucleus to the cytosol. Therefore, it is not present to bind to the origin recognition complex. ORC2, one of the subunits of the origin recognition complex, is phosphorylated, which reduces its affinity for chromatin. ORC1, the largest ORC subunit, is phosphorylated, which results in its degradation. Without these two subunits, the origin recognition complex is unable to form at replication origins. Also, CDT1, the protein that brings the MCM complex to the origin, is phosphorylated, which promotes its degradation. A protein called geminin is expressed in S phase, which binds to any remaining CDT1 and prevents it from interacting with the MCM complex. Note that geminin stays in the cell through G2 phase and M phase and is only degraded during G1 phase. So as the cell starts synthesizing CDT1 again after S phase is over, geminin is still present to prevent CDT1 from bringing MCM proteins to the origin. Only in G1 phase is geminin itself degraded, which frees CDT1 for interaction with the MCM complex. These redundant mechanisms all contribute to preventing replication origins from being licensed during S phase. This may seem like overkill, but having multiple independent mechanisms makes control of this process that much more complete. If replication began more than once at even one replication origin during the cell cycle, that would lead to chromosomal abnormalities, which can be very harmful to cell function. So this concludes my consideration of DNA replication. I've covered the basic process of replication, how supercoiling is created and dealt with, and the process and regulation of initiation in bacteria and eukaryotes.